Dear students, this is Santosh, lecturer in biology, and usually I teach biology for PUC 1 and PUC 2 for board exam as well as for NEET exam. And today I am going to deal with a topic, regulation of gene expression. It is from 2nd PUC syllabus, the chapter molecular basis of inheritance. And the students who completed 2nd PUC know very well that this is the most important topic. There would be a definite question whether it is NEET exam or state level CET exams. And students, today let us understand what exactly regulation of gene expression is. The mechanism behind the regulation of gene expression was explained with a Nobel Prize winning concept which is called lac operon concept by the two Nobel laureates Jacob and Monod. And students, to explain the mechanism behind the regulation of gene expression, Jacob and Monod studied the metabolism of lactose in E. coli bacterial cells. By studying the metabolism of lactose in E. coli bacterial cells, Jacob and Monod explained the mechanism behind the regulation of gene expression, for which they won the Nobel Prize. And students, as you all know that whenever an organism takes birth, it takes birth with its complete DNA, and that DNA comprises of hundreds to thousands of genes. But it is very clear that no organism requires expression of all the genes all the time. So with this, it is very clear, whenever an organism requires the expression of a particular gene, then only that gene is said to be switched on and it starts giving its expression, and mostly that expression is nothing but a protein. And whenever that protein is not required to an organism, that gene is said to be switched off and that protein is not produced. And what is that mechanism? Turns on the gene expression and turns off the gene expression was given by Jacob and Monod, as I already said, by studying the lactose metabolism in E. coli bacterial cells. And students, I told you the concept is lac operon. So here you should understand, in lac operon, lac stands for lactose and next word is operon. And let us understand what is a operon. And students, I already told you that operon is a genetic unit, or in very simple terms, operon is nothing but a small fragment of DNA, and any fragment of DNA naturally contains genes. And now let us see the definition for an operon. This is the standard definition. Operon is a genetic unit, contains one or more structural genes, and adjacent to it are control genes. So operon is a small fragment of DNA. In that fragment of DNA, what are present? Structural genes are present, and adjacent to it, control genes are present. And the operon studied by Jacob and Monod, it contains total 6 genes, out of which 3 are the structural genes and remaining 3 are the control genes. And now let us see the functions of 3 structural genes of lac operon. It is clearly written, concentrate on this. Structural genes of lac operon are, first one, cistron Z. Already I told you, structural gene also can be called as cistron. So cistron Z, what is the function of cistron Z? It codes for an enzyme called beta galactosidase. And what is the function of beta galactosidase? It always splits the lactose into glucose plus galactose. And the next structural gene is cistron Y. It codes for an enzyme called permease, which promotes the uptake of lactose by the E. coli bacterial cells. Means whenever we add lactose to the nutrient medium, permease increases the permeability of cell walls of bacterial cells, and more lactose is taken into the bacterial cells with the help of this enzyme called permease. And third cistron is cistron A, and whose function is not related to the lactose metabolism. So in this way, in total lac operon, total number of structural genes is 3. Structural gene is otherwise called cistron, and the 3 cistrons are cistron Z, cistron Y, and cistron A. And 3 control genes are named as R, P, and O. R for regulator. What is the function of regulator? It codes for a protein called repressor. P for promoter. It is the gene which acts as binding site for the enzyme RNA polymerase. O for operator, and operator is very important because it acts as on-off switch for the entire lac operon, because it is the binding site for the repressor protein. So this is the structure of lac operon, in which total 6 genes are present. Out of 6 genes, 3 are the structural genes and 3 are the control genes. Dear students, till now I have explained you the structure of lac operon, and this is the diagrammatic representation of lac operon given by Jacob and Monod. I already told you lac operon is a small fragment of DNA. You can see in the diagram, here is a fragment of DNA. In this fragment of DNA, total how many genes are present? It's very clear. Total 6 genes are present, that is 1, 2, 3, 4, 5 and 6. And out of these 6 genes, and here it is clearly written, R, P and O together called control genes, because they control the expression of the structural genes. So what are the 3 control genes present in lac operon? R for regulator, P for promoter and O for operator. And functions we have already studied. And in the diagram you can see, adjacent to the 3 control genes are present 3 structural genes. Structural gene is also called a cistron. And students, remember, if you ask me what is a cistron, in very simple term, cistron is a gene which specifies a protein or which codes for a polypeptide. The gene which codes for a polypeptide is called a cistron. It is otherwise called a structural gene. And in lac operon of Jacob and Monod, how many structural genes are present? 3 structural genes are present. That is cistron Z, cistron Y and cistron A. And what is the function of cistron Z, Y and A also we have studied. So always remember, this is the structure of a lac operon. It is a fragment of DNA in which totally 6 genes are present. Out of 6 genes, 3 are the control genes. Which 3 are the control genes? Regulator, promoter and operator. Adjacent to control genes are present structural genes. Which 3 are the structural genes? Z, Y and A. Structural genes are otherwise called cistrons. Dear students, till now we understood the detailed structure of lac operon. Now let us understand the mechanism of lac operon, or how this lac operon regulates the gene expression. For our study convenience, mechanism of lac operon can be divided into case 1 and case 2. Now let us go with the case 1. Case 1 is otherwise called switch off mechanism, or it is the mechanism without lactose in the nutrient medium. So let me repeat. To understand the mechanism, we can classify the mechanism into case 1 and case 2. So my case 1 is commonly called switch off mechanism. Why it is said to be switch off mechanism? During this case, E. coli bacterial cells were grown on the nutrient medium without lactose. And already we are familiar with the structure of lac operon. Now you can see here, I have drawn the lac operon containing 6 genes. You already know that there are 3 control genes. What are the control genes? Regulator, promoter, operator, and the 3 cisterns are Z, Y and A. I already told you the function of regulator gene, it always codes for a protein called repressor. So where is the binding site of repressor? Repressor protein's binding site is always present on operator. Due to the absence of lactose in the nutrient medium, immediately after the production, repressor protein moves towards the operator and binds with the operator. And students should remember, binding of repressor and operator always follows a lock and key mechanism. Means if you imagine operator as a lock, repressor is a key, so that you can fix this key into this lock only. Due to the absence of lactose in the nutrient medium, what happened, tell me? Regulator gene coded for a protein called repressor. Repressor protein being having its binding site on operator, it went and attached to the operator, and now the operator is said to be switched off. That's why we call this mechanism as switch off mechanism. And I already told you that adjacent to operator, which control gene is present? Promoter is present. And promoter is the binding site for the enzyme RNA polymerase. Already I told you this. And students, there are 3 cisterns here, that is Z, Y and A. Actually they code for enzymes, I said, but you know all enzymes are proteins. If the protein has to be produced from a gene, first that gene should participate in transcription. And for transcription, which enzyme is very important and essential? RNA polymerase enzyme is essential. But now you see, for RNA polymerase enzyme, operator acts as a roadblock. What is the reason? For RNA polymerase enzyme, operator acts as a roadblock because repressor already got attached to the operator. So RNA polymerase cannot cross the operator. As it cannot cross the operator, the 3 structural genes, that is Z, Y and A, cannot participate in transcription. That's why I have written here, no transcript is formed, because transcription is not taking place. Why the transcription is not taking place? Because RNA polymerase enzyme is unable to cross the operator. Without enzyme there is no transcription. No transcription means no mRNA is produced, and without mRNA there will be no translation, and without translation enzymes cannot be produced. That's why I have written no enzyme. So students should understand, though 3 structural genes are present which actually can code for 3 different enzymes, but in case 1, that is in switch off mechanism, They are unable to code for the enzymes. Means 3 structural genes are not giving their expression in case 1, which is called switch off mechanism due to the absence of lactose in the nutrient medium. Dear students, we understood case 1 mechanism. I already told you it is called switch off mechanism. Their nutrient medium was without lactose. Now, coming up with case 2, and here you can see case 2 is always called switch on mechanism because now the nutrient medium is having lactose. So simply students can remember case 2 mechanism of lac operon. It is otherwise called switch on mechanism because it is the nutrient medium with the lactose. And let us see what exactly switch on mechanism is. Jacob and Monod has cultured the E. coli bacterial cells, and for that E. coli bacterial cells, now they have prepared a nutrient medium, and that nutrient medium contains lactose now. So that's why let me represent lactose here. And already you know whether you add lactose to the nutrient medium or not. What is the function of regulator gene? It always codes for a protein called repressor. And I already told you repressor's binding site is always present on what? Operator. And it also follows lock and key mechanism, I said. But now you can see in the diagram repressor, this is the repressor protein. Immediately after the production, repressor is naturally trying to move towards its binding site, that is operator. But who blocks the repressor now? I already told you nutrient medium is having lactose. Let me show lactose here. Lactose binds to the repressor and lactose blocks the repressor. When lactose blocks the repressor, repressor fails to bind with operator. Now, the operator is said to be switched on. If you remember in case 1 mechanism, when repressor binds with operator, operator was said to be switched off. But now repressor is unable to bind with the operator. Why repressor is unable to bind with the operator? Because lactose blocks the repressor, so that repressor fails to bind with the operator, and now the operator is said to be switched on. So you can see here this is promoter, and on promoter which enzyme is present? RNA polymerase enzyme is present. Now RNA polymerase enzyme can easily and happily crosses the operator because operator is on. As RNA polymerase enzyme crosses the operator, the 3 structural genes are transcribed. Means RNA polymerase enzyme performs transcription of the 3 structural genes, and what is produced? A transcript is produced. A transcription is happened. A transcript is produced, or simply I can say an mRNA can produced, and this mRNA is a polycistronic mRNA because we are talking about prokaryotes. In prokaryotes, single mRNA carries the genetic information of more than one cistron, and such mRNA is called polycistronic mRNA. Means for 3 cistrons, 3 mRNAs are not produced. Only one mRNA is produced. That's why it is called polycistronic mRNA. This polycistronic mRNA participates in translation, and it results in the formation of what? All the 3 enzymes. In case 1, if you remember, structural genes were present, but structural genes were not transcribed, so no enzymes were produced. Hence we call that mechanism as what? Switch off mechanism. And now I am telling you case 2, and this mechanism is called what? Switch on mechanism. As the structural genes are present, structural genes participate in transcription, polycistronic mRNA is produced, and all the 3 enzymes are also produced. So with this, I want to conclude both in case 1 and case 2, structural genes were present. But in case 1, structural genes were unable to give their expression, and in case 2, structural genes has given their expression. What is the main difference between case 1 and case 2? Presence or absence of lactose. So in case 1 there was no lactose, no enzymes were produced. In case 2, nutrient medium contains lactose, hence the enzymes are produced. So ultimately, which substance is acting as the inducer? Lactose is acting as the inducer. So that's why lac operon is an example for inducible operon, and lactose acts as the inducer. This was the conclusion given by Jacob and Monod by studying lactose metabolism in E. coli bacterial cells, by doing which they have explained regulation of gene expression in prokaryotes, and for which they awarded the Nobel Prize. Thank you.